So in this section is very interesting because we're going to explore, other than carbohydrate, right? How does protein and lipids also are able to give us energy? So let's look at the learning outcome first. Explain the relative energy values of carbohydrate, lipids, and protein as respiratory substrates. So in order to know the energy value, we need to know how they're being respired. Then only we know. So let's start. So all these previous lessons we can skip already. So today we're going to look at alternative substrates for aerobic respiration. Alternative means other than C6H12O6 that I've learned. Okay. So and then we're going to look at, so what are they? Protein and lipids. So we're going to look at entrance of these alternative substrates to respiratory pathway. So how do they enter our respiratory pathway? Where is the point of entrance? Okay. And then that they all share the common pathway. So that means to say that we already learned the respiratory pathway, right? Glycolysis, link reaction, Krebs cycle, oxidative phosphorylation. And then our protein and lipids also come and enter the same common pathway, but the point of entrance is different from glucose. So where do they enter? Okay. So because they share the common pathway, we save our enzyme because we only produce one set of enzyme. We can use it for all the substrates compared to after lipid, we have to produce another set, and then protein will produce another set. Then we were wasting our amino acids and ATP to produce them. So now we share all of them, so it's cost effective. So we're going to later explore the energy value of carbo, lipids, and proteins. And after you explore, you will realize that why is it that the H2O ratio or the percentage of H atom, or can you please add another one in, which is actually the, or that C-H is the main determinant energy of the respiratory substrate. EJ, please look at my screen. My screen? Yes, my screen? Yeah. Yes, good. Okay. Please add C-H to your notes, please. Okay, right. H2O ratio is there already. Okay, right. So how do we obtain energy from the food? So let's start with the food. Food contains carbo, lipids, and proteins. So let's start with carbohydrate first. How do you obtain energy from carbohydrate? Is by respiration, right? So carbohydrate is the first and the main respiratory substrate. Why? Because all the cells in the body can use carbohydrate for respiration. Like brain cells, neurons are nerve cells, red blood cell, lymphocytes. These cells, right, can only use carbohydrate for respiration. That's why carbohydrate is the main respiratory substrate. Now, if you want to talk about red blood cell, why red blood cell can only use glucose? Because red blood cell got no mitochondrion, right? It can only carry out only glycolysis. It can carry out. And what enters glycolysis? Glucose, right? The very first step is glucose, right? Then you activate the glucose. So that's why red blood cell can only use carbohydrate as a respiratory substrate. Okay, and as an example. Now, when our glucose is in short supply, when we don't have enough glucose, we start to actually use lipids. Why not enough glucose? Because of prolonged starvation, or when a person go for diet, they don't take carb, no carb, no carb, no carb. For many weeks also no carb. After that, they start burning their fats. Then you can start to see they shrink already. Okay, so the fats are stored in the liver and adipose tissues, and also in the seeds for plants. Okay, then for proteins is used as a last resort. Can you please change this resource to resort, please? R-E-S-O-R-T, resort. Okay, last resort means you got no choice already. You got no more help, so you have to get the help from protein. Okay. So protein is used as a last resort when fats run low. That means to say no more carbohydrate because you're not eating anything. And then you start to burn the fat, also fat no more already. Then they start to burn your muscle. So therefore, this must be the last one to be burned because proteins are so important for us to make our muscles for movement, locomotion, and then to run away from danger, for hunting, for doing work. Last time it's hunting, now no hunting, now it's doing work. Okay. So therefore, protein is the last one because it's actually burning down your muscle, right? Okay, so now let's look at carbohydrate. How do we get energy from carbohydrate? Carbohydrate is hydrolyzed in our digestive tract, okay, and it becomes hexose sugar like glucose. Then it enters the respiratory pathway. So start with glycolysis, okay, to form pyruvate. After that, you got link reaction, we form acetyl-CoA. After that, got Krebs cycle. This is the respiratory pathway, right? Of course, you've got oxidative phosphorylation. What about lipids? So how does lipid enter into our main respiratory pathway? Lipids in our digestive tract will be hydrolyzed into fatty acid and glycerol. Now, glycerol, because it has 3 carbon, so it will enter pathway as triose phosphate. It will be converted to triose phosphate and enter the pathway here. And then fatty acid, right? Fatty acid has got a lot of fatty acid tail, very long. So it's actually fatty acid is what? COOR, okay. Yeah.
What is the
Health. So it's got this fatty acid tail here, right? Fatty acid got a lot, the long fatty acid tail. And then got a C double O bond. Okay. And then where's my COOH? So this is O here, and this is OH here. Where's my OH? So this is my fatty acid, right? So it got very long hydrocarbon tail, So therefore, how to enter respiratory pathway is that there's an enzyme that will chop off the tail 2 carbon at a time. Okay, so chop. Use red color better. So chop here, chop here, chop here. This is 1, 2, chop here. Okay, so it will chop the fatty acid tail 2 carbon at a time, and this 2 carbon becomes acetal and enters the respiratory pathway. So this process is actually called the beta oxidation, where they remove 2 carbon at a time from the tail of the fatty acid. Okay, so what about protein? Protein is hydrolyzed in a respiratory pathway, hydrolyzed in a digestive tract to become amino acids. And then these amino acids, right, in order to enter the respiratory pathway. Now, all the substrates, intermediate substrates of respiration, they have no nitrogen one. Got not? Don't have, right? So therefore, but amino acids got nitrogen because amino acid is this thing here. You got the central carbon, right? And then to the right is acid. Okay, to the right is acid. And to the left is actually the NH2, right? Okay, yeah, look. Where is it? NH2, H2N dash, correct? And then you got the central carbon, got the R on top, and then got the H at the bottom. So this is your amino acid. Hello, where are you? Okay, so in order to enter the respiratory pathway, we have to remove this amine group which contain nitrogen. So therefore, the first step involved is deamination to remove the NH2 group. So deamination will remove the NH2 group, so become ammonia, and then the carbon skeleton is the keto acid. The remainder carbon skeleton is the keto acid. So keto acid is now ready to enter the respiratory pathway because no more NH3. So it will enter respiratory pathway depending on the number of carbon that it has. Because different amino acid got different R group got different number of carbon. So if it's a 3 carbon, it will enter as pyruvate, 4 carbon as oxaloacetate, 5 carbon as alpha ketoglutarate. Okay, so by now you need to remember 6 carbon is citrate, 5 carbon is alpha ketoglutarate, 4 carbon is oxaloacetate. You need to memorize.
Oh my God.
Don't ask me. You write down on the paper, stick it to your toilet, stick it to your laptop, stick it to your car, stick it everywhere that you use every day until you remember. Then only you unstick.
Oh, can you just write so we know how to spell it?
No, you check your notes. Okay, next.
Wow.
Yeah, because I got no time to repeat something that is already given the notes. Okay, I have to cover the syllabus. So go back to your notes and check. Okay. Why? Okay, why common respiratory pathway already mentioned? Because they share the enzymes. So the same enzymes can be only synthesized one and reused again. So it's more cost effective, use less ATP and less raw material, means amino acids. Okay, now there's a table for you to fill in the blank. So this table is for you to understand what governs the energy value of our respiratory substrate. So for carbo, lipid, and protein, I'm taking an example so they can do calculation. So I'm using the monomer glucose, lipid, stearic acid, protein, glycerin to calculate the energy value to represent carbolipid protein. So I also give you the formula of these three molecules. So what you need to do is that you need to calculate the rest of the three rows. So I give you 1 minute to calculate now. 1 minute. Okay. So please calculate now. Okay, whatever. Right. So let's look at the answer. So you can see that over here, after calculating the total hydrogen per mass of substrate, in percentage, right? Yeah. Lipid got 11.8% of hydrogen per unit mass. Carbo 6.7 and protein 7.9. Does it tally with the energy value? Does it tally with energy value? Tally means you know that, okay, lipids produce twice amount of energy compared to carbohydrate. Does the hydrogen, percentage hydrogen also tally 2 to 1? Yes. Can you see that? Almost there, right? So what is the conclusion we can make from this calculation is, this one sometimes you can be asked in exam. They ask you to calculate and then they give you a table, they ask you, so what conclusion can you make from the results? So you just have to say, what can you see from result? Oh, I can see that lipid, energy value of lipid, sorry, not. Yeah, in your calculations, hydrogen per mass of substrate is higher in lipid compared to protein, compared to carbohydrate.
Yeah, but it's not fair. The lipid got so many carbons. Only got 6 carbons.
But the ratio is still greater.
Yeah. So therefore, the energy value, you see now because of the hydrogen, percentage hydrogen, right, per mass, the energy value is also the same. Lipid more than protein, more than carbohydrate per unit mass. Okay. Now, what do you mean by energy value? What is that? How do you calculate? How do you represent energy density? Energy density is the energy per unit mass. Okay. Kilojoule per gram is energy density. So what determines the energy value of a substrate? Now you already know that it is actually the number of atoms per unit mass of the molecule that determine the energy. And you can further actually explain why it's that hydrogen. Because you have learned that, okay, the substrate that you learn is C6H12O6, right? So why is it not carbon determine the energy value? Why is it hydrogen? Why is it not oxygen? Why must be hydrogen? Because this molecule only got either carbon or hydrogen, oxygen. Why is it is the hydrogen that determine the energy value? Why not carbon? Why not oxygen? Why hydrogen? Very good, Martin. Okay. Okay. Why is it the hydrogen or H2O ratio that determine the energy value? A good try, EJ. Okay. Now, the more reduced, the higher energy. This one is, it means the more hydrogen, reduced means the more hydrogen substrate has, the more energy. Why? Because you know that during respiration, you already learned, during respiration, right, it's either decarboxylation, remove the carbon, or dehydrogenation means remove the hydrogen. It's either or. And you know that when you remove the carbon, you produce CO2. Where got energy? No energy, right? CO2, no energy. So the only part that can give you energy is the hydrogen already. And this hydrogen is 2 hydrogen being removed at a time, will always be carried by the hydrogen carriers. And the hydrogen carriers are what? Either the NAD inwards or FAD. And when they carry, they become NADH, NADH plus H plus, and FADH plus H plus. And then finally, they carry them and release it to oxidative phosphorylation. Oxidative phosphorylation. So oxidative means when the carriers are being oxidized, right? They release the hydrogen, right? They're being oxidized. That will lead to phosphorylation, means ATP synthesized by chemiosmosis. So that is why it is the hydrogen of the substrate for respiration that determine the energy value, because these are the hydrogen that's being collected, right? Starting from glycolysis to link reaction to Krebs cycle, collect all the way ready, and finally bring to oxidative phosphorylation and oxidize it completely with oxygen to produce ATP. How many ATP per glucose? From oxidative phosphorylation per glucose? No, oxidative phosphorylation only. 34, very good. Because 4 is substrate level phosphorylation. Yay!
Wait, explain again.
Never mind, go back and revise yourself.
Wait, I'm so lost. No. Oxidative phosphorylation. What?
Go back to the slide and check, okay? You got substrate level phosphorylation, oxidative phosphorylation. Substrate level phosphorylation give you 4.
Okay.
So 38 minus 4, how much?
34.
34 from oxidative phosphorylation. Okay, finish. So why? Hydrogen atoms will be carried by NAD and FAD, okay, in the form of reduced NAD, reduced FAD, goes to oxidative phosphorylation to produce ATP. So more hydrogen atoms, the more hydrogen atoms the substrate got, the more NADH and FADH, the more oxidative phosphorylation, the more ATP. So hydrogen is the main determinant of energy value of a substrate. Yeah.
You have to explain an example of why hydrogen is used.
Yes. Every point here is 1 mark in exam. Okay, next. Of course, if you don't have to explain, I won't write in my PowerPoint anyway. Especially highlight means keywords. Okay, it's easy. EJ, it's very easy. You understand, you can write. Okay. Now, this is a table of composition and fuel values of some common foods. So we are only interested in carbo, fat, and protein to show that the fuel value, remember, kilojoule per gram is energy density. Remember, kilojoule per gram is energy density. So fat 38, carbo, protein 17. So fat has twice the amount of energy compared to carbo and protein per gram of them. Okay. So how do you determine the energy value of these molecules? You can actually oxidize them in a calorimeter. So you measure a known mass of the substrate, put it here, and then fully oxidize it. And then the heat will be generated. So when it oxidizes, a lot of heat will be released. This heat will heat up the water, a fixed volume of water, to raise the temperature. So you can use a formula to calculate how much heat is needed to raise the temperature of this water by certain degrees Celsius. Okay? So that's how you find out the energy value of the molecule. Okay. So in this lesson, you have learned alternative substrate for aerobic respiration, and then the entrance of all these alternative substrate into respiratory pathway. Where do they enter the point of entrance, and that they share the common pathway. And the advantage of sharing is that it is more cost effective because you only produce the enzymes for the common pathway. And you also look at the energy value of lipids is more than proteins, which is more than or equals to carbohydrate, depending on the combination of amino acids and the R groups of the proteins. Okay. And the H or H to O ratio is the main determinant of energy value of a respiratory substrate. Okay. So that's all for this section here.